Please respond.
Sorry, sir, I'm not attended that class. I don't know, sir.
Sangeeta Venkateshwaramma. Did you attend yesterday?
Yes, sir.
Tell me, what is the last class? Yesterday class. What we completed yesterday? Translation in eukaryotes. Gene expression in eukaryotes, right?
Yes, sir. Eukaryotic translation, sir.
Eukaryotic translation and gene expression in translation.
Yes, sir.
Can you explain in brief about the translation? What is translation first? What is translation? Translation means from mRNA strand, from mRNA polypeptide chains prepared. This process called as translation. Then of the formation of mRNA strand, mRNA strand prepared from DNA by the process of transcription, right? What we completed yesterday, initially we completed the transcription process. Transcription means from the DNA, mRNA prepared on the mRNA strand, polypeptide chain prepared. This polypeptide chain called as protein. So the first process, DNA to mRNA transcription. Transcription. Why we explain? Why I am explaining again this transcription translation, because the same concept would be repeated in PCR contest also, in PCR lesson also. So from the DNA to mRNA transcription process, here also 3 stages: initiation, elongation, termination. And after formation mRNA, it is not full matured mRNA initially. After post changes such as methyl capping formation, removal of introns, and polyadenine A. So due to these changes, it fully matured. After that, this is transported to cytoplasm, where they started translation process. In translation also, the same steps are involved in this. Before that, we should know about what are the requirements from the translation. The main requirements are mRNA strand already prepared, amino acids, ribosomes, tRNA, they are all involved. In first step, amino acids activated along with tRNA and form amino acid tRNA complex. Second step, 43S complex formation. And next one, scanning and adding of large subunit. adding large subunit. Next, this is only initiation process of translation. So after initiation, what is the next step?
Elongation.
Elongation of what elongation here? How elongation started? Elongated started the some tRNA with amino acid, second amino acid enter into the A site. Now translocation occurs and it would be continued. And final step, during translocation and elongation process, what are prepared on the mRNA stand? What are prepared? What are synthesized? During elongation. And you should remember the initiation factors and elongation factors too. No one attended yesterday. Hello? Nishat. Nishat. Jnaneshwari.
Sir.
Please respond.
Yes, sir.
During elongation stage, what are prepared on mRNA stand? What are synthesized?
During elongation.
Yes, during elongation, what are prepared on mRNA stand? Can you explain in brief about elongation?
Amino acid 2 plus.
Yes, amino acids.
To the A site.
Right, A site. Next,
Then peptide bond formation.
bond formation between.
Between amino acid 1 and amino acid 2.
Right, amino acid 1 and amino acid 2. Which bond formation between these?
Peptide bond is formed.
Peptide bond formation, right. And
Then
next,
translocation.
yes, translocation. Next,
In translocation, translocation of ribosome, ATP is required.
okay.
And elongation factor 2 is also required.
Okay.
In translocation, that amino acid 1, it is on the P side, which is transferred to the E site.
Okay,
and
right.
Amino acid 2 is shifted to, which is on A side, which is shifted to P side.
Right. Then which place empty?
Now, P side is empty.
P side means peptide.
No, A side is empty, sir.
Right. A side is empty. Now the elongation continues until elongation continued until reach termination codon. When the P side reach the termination codons UAA, UGE, UAG, when reach the P side, exactly these three codons, nonsense codons, immediately the termination factor initiated are binding them. Right?
Yes, sir.
Now the polypeptide chain terminated. Now this protein strand prepared.
Yes.
And next another topic, yesterday we completed the gene expression. What is gene expression? What express the gene?
Gene.
Jnaneshwari. Srisailam. Srisailam. Hello? Right. Okay, gene expression.
The
Hmm.
the creation of proteins from its gene is called gene expression.
Right. This is also gene expression here. Thus, this is gene DNA. This is gene DNA. Due to the transcription translation, the protein stand prepared. This protein enzyme act on this gene substrate. The gene substrate. When this protein act and react on the gene substrate, then only the character express. It is the gene expression, nothing but character express. Yesterday I explained very clearly with examples also. Right. So this is gene expression concept. In gene expression also we completed gene banking, gene battery and chromatin changes and some restriction enzyme changes. They are all we completed yesterday. Now today class is the most important one, restriction endonuclease. Restriction endonuclease. Right. The main function of restriction endonuclease recognize the specific site and cuts the DNA fragments and also modify the DNA. So mainly three changes. Each and every restriction nucleases first recognize, next cleavage or cut the DNA strand at appropriate site, and next it can modify the DNA sequence. It can modify the DNA also. It can modify the DNA strand also. So this is the major functions of the restriction endonuclease. Let's see the types of restriction endonuclease. Normally there are three types of restriction endonuclease. The first one, restriction endonuclease. Type 1 endonuclease, type 2 endonuclease, type 3 endonuclease. Restriction endonuclease is important for short answer. Okay, this is very, very important. Let's see the first one. Already told you the main concept of restriction endonuclease: recognize, cleavage, modify. Right. First type 1 restriction enzymes, which is complex structure with multiple subunit. Complex structure and with multiple subunit, and it can modify the enzymes also. They mainly this type of enzyme, type 1 enzyme extracted from the E. coli, Escherichia coli bacteria. The properties of this enzyme cut at a site that differs and in some distance at least 100 base pairs away from their recognition site. Means, for example, this is the DNA strand, this is recognition strand. This from recognition site and 1000 base pairs, 100, 200, 300, 500, for example, these are the 1000 base pairs. At the region of 1000 base pairs, this is recognized site, this is 1000 base pair number. The 1000 base pair number cleavage occur. 1000 base pair number cleavage occur. 1000 base pair cleavage occur. Cleavage occur. Now at this site, 8,000 base pair site cleavage occurred. First of all, here the recognition site different and cleavage site different in type 1. Right. In type 1 recognition, this is recognition. This is cleavage site. Cut the DNA. So recognized site should be composed of 2 portions. It may be 3 to 4 nucleotides or 4 to 5 nucleotides or 6 to 8 nucleotide. Means, for example, this is the recognition site. In recognition site, the number of nucleotides may be 4 to 5, or 3 to 4 nucleotides, 6 to 8 nucleotides. So that is a depend on the species. So maybe the recognition site number of nucleotides may be 3 to 4, 4 to 5, 6 to 8. And for this type 1 enzyme activity should require magnesium ion and also ATP energy. For this cleavage reaction, for type 1 restriction enzyme, required magnesium and also ATP energy. ATP energy. And the type 1 restriction enzyme possesses several subunits. Already told you the multiple subunit. At the very beginning, tell you this is the complex and the restriction type 1 enzyme multiple subunits such as HSDRA, HSDR, for example, for suppose this is the type 1 restriction endonuclease. So this main subunits 1, 2, 3, 4 or 5. So first subunit HSDR, HSDM subunit, HSDS, and again HSDR. So these different type of subunits. But here mainly HSDS is the important for the specificity to cut the recognition, cut the site recognition. In addition, its methylation, methylopherase activity. Already told you methylopherase activity, methylation yesterday. What is methylation? Hello? What is methylation? Hello? What is methylation activity? What is methylase activity? What is acetylation? Hello?
Yes, sir.
What is methylation?
Activation
Epigenetic control, epigenetic gene control. The clearly explained about methylation, acetylation, deacetylation. What is methylation?
of epigenetic gene control.
How it would be controlled? Methylation activity means the methyl group added to the methyl group added to the cytosine of 5th carbon. Right?
Yes, sir.
Now the chromatin, the more and more become complex. As a result, the gene expression prohibited. That means transcription prohibited due to the activity of methylation. Right?
Yes.
Next one. This is type 1 restriction endonuclease. Right?
Yes.
Now type 2. Type 2 restriction endonuclease. The type 2 restriction endonuclease differ from the type 1. Mainly it is a multi-enzyme complex. The first of all, the type 1 enzyme only the complex enzyme with multiple subunit. But inside the enzyme multiple enzyme complex. That means the two functions carried out. One is recognition, modification. Second one, cleavage enzyme. And another important one, when compared with type 1 recognition site different and cleavage site different. But type 2, the recognition site, after recognition, the site itself cleavage takes place at the site of recognition. That is the main concept of the restriction to endonucleases. For example, this is the DNA strand. This is recognition site with 2. This is recognized. But in this side also the cleavage takes place. So that is the multiple function. Recognition with the same enzyme and the cleavage with the same enzyme. That is the multiple. And here important thing is the same like type 1 also, the recognition site should be 4 to 8 nucleotide in length. They recognize and cleave the DNA at the same site. This is important. They recognize and cleave the DNA at the same site. And another most important, they do not use ATP, but required only Mg2 plus as a cofactor. What about type 1? Hello?
Yes.
Mg plus and ATP energy.
Both are required.
Yes.
ATP energy required
Yes, sir.
and also Mg2 plus cofactor required. But in 2, only cofactor required. No use of ATP energy. And this type of enzyme is mostly useful in rDNA technology. So that's why this is the very, very important enzyme in molecular biology or rDNA technology. Second one, next one, third, type 3 restriction endonuclease. This is single enzyme, not a complex, single enzyme. This type 2 restriction enzyme recognizes 2 separate non-palindromic sequence and the cut the DNA from the recognition site away from 20 to 30 base pairs. Type 3. Type 3. This type 3 enzyme recognizes to separate non-palindromic sequence. What is palindromic? Non-palindromic. The palindromic means forward direction, reverse direction, same sequence. That is palindrome. The mainly restriction 3 enzyme recognize non-palindromic sequence. And another important one is, for example, this is the DNA strand. This is recognition site. From the recognized site, here 20 to 30 base pairs. Someone who raised the hand. Hello? Any doubt?
Yes, sir. Can you please tell me what is palindromic?
Right. Palindromic means G A A T T C. This is, for example, 5n to 3n direction. Right? Can you write the complementary sequence to this? Can you write complementary sequence? Tell me.
C,
Right, C,
T,
T, okay.
T,
Right.
A,
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 G.
right. Now first, actually these two stands. Let me draw with different colors. One is red color stand. Another one is blue color stand, right? Okay. The first you read the red color from one direction. I will show the direction, right? This direction, forward direction, that is forward direction. Read forward direction sequence. Hello.
G, A,
G A A T T C, right?
yes.
Now read the backward direction to second one.
Second one, G, A, T, T, 6. Okay, is it, it's same like
Right.
vice versa.
That's it. The same sequence repeated forward backward. That is palindromic sequence. Okay.
Okay, if it's dissimilar, it is non-palindromic, right?
Non-palindromic, right? Non-palindromic.
Okay, sir. Thank you.
Right. Right. Okay. And another important is recognition site and away from this cleavage takes place away from the recognition site. Now just compare the 1 and 2 and 3. The main in the first one recognition site away from 1000 nitrogen bases cleavage occur, right? Hello?
Yes, sir.
Yes, sir.
And another one is in for cleavage activity, the first for type 1 endonuclease requires ATP energy and
Magnesium.
magnesium cofactor also.
Yes, sir.
And what about the second where the recognition site present, then only the cleavage also takes place and only magnesium cofactor required without ATP energy.
Yes, sir.
And the third one, recognition site away from the 20 to 30 base frame upstream. Upstream, do you know upstream and downstream? For this is recognition site, this is upstream, and this is downstream. And these enzymes contain more than one subunits and require ATP. Require ATP and also cofactor magnesium also required. This is the type 3 completely type 1. The only one different is a 20 to 30 base pair away cleavage takes place, but in first 1000 base pair away. That is the main difference, right? Is it clear?
Yes, sir.
Okay, remember one thing, almost all restriction enzyme cleave at the palindromic sequence. Cleave or recognize that the palindromic, where the palindromic sequence are present, then there only the cleavage take place, right? But in only third one also palindromic and cleavage and non-palindromic also cleavage. I will tell you later how the non-palindromic cleavage takes place and how the palindromic cleavage takes place. Okay, right? And someone writes the exam 245063.
Okay, sir.
Who is 245063?
Kavita.
Kavita, any doubt?
No, sir, no.
Okay, right. Now nomenclature. Nomenclature. The nomenclature of a restriction enzyme, for example, the abbreviation of restriction endonuclease 1, ECORI. This is the abbreviation of endonuclease 1, restriction endonuclease type 1. So the nomenclature, first word refers, this first word refers genus, and this second two species, and third one strain, bacteria strain. Bacteria, different type of strains are there, from which strain the genome isolated, the based on that given the strain name also. And fourth one order of identification. Order of identification. Order of identification. The first one, order told you the restriction endonuclease 1 type 1 mainly discovered in Escherichia coli bacteria, right? Hello?
Yes, sir.
Escherichia coli bacteria. That's why the brief abbreviation name ECORI. As I suggested, first E for genus. What is the genus name here? E for
Escherichia.
genus. Escherichia.
Escherichia.
Right. Escherichia. What is the next word? Two species. Coli is the species. And what is the third one strain? Strain type R strain. R13 strain. Order of identification, first order. So this is first time, that's why given first one, number one. So this is the nomenclature of ECORI, right? Next one, for example, bomb HI. Is it clear first?
Yes,
Hello?
sir.
Right. Actually, in our book given only one enzyme ECORI, and you have to add two more things. The first one, bomb HI restriction enzyme. Is it clear? Can I rough it?
Yes, sir.
Another nomenclature, BAMH here also same one. H1 order one. B for first, this is indicate
Genus.
genus. AM for these two words,
Species.
species. Sorry. Please be silent. Third one, strain. Order of identification. Order of identification. Right? So here also same first, genus, B for Bacillus. Bacillus. Species AM for amyloli, quiescence, amyloquiescence for AM, and strain name is H strain. Same here also order 1. Here also order 1. So this is another type of nomenclature. And third one, this is second one, bomb H1, and third one hind 3. Third one hind 3. Is it clear? Second one, bomb H1 nomenclature. Hello?
Yes, sir.
Right. For example, hind 3, serial number 3, HIND, hind 3.
In BAM HI, sir, please. In BAM HI, what is the H, sir? Strain name?
Strain name H.
Strain name only H. Okay, only H.
H.
Sir, species name, sir?
What?
Amyloliquidation.
Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. AM amylo.
Yes, sir.
Thank you,
Okay.
sir.
Okay, sir. Thank you.
And another one, hind 3. First word. Tell me, first one, H.
Genus.
Genus. And the second 2 words, EN. Species. Strain. Number. Order of identification. That means identification number. Right? So hind means H for Haemophilus. Write down this, Haemophilus. Species IN for influenza. Strain D. And recognition number 3. So this is hind 3. This 3 enough for nomenclature. Of course, in your book given only 1, but you have to add 2 more. Okay?
Yes, sir.
Is it clear?
Yes, sir.
Right. A nomenclature of endonuclease enzyme also important. This is also short answer. Okay?
Yes, sir.
Right.
Sir, it's order number 3.
Yes. Order number 3. Next one, recognition site. Recognition site. Already told you restriction enzymes recognize the specific sequence of nucleotide and produce a double stranded cut in the DNA. And recognition sequence vary between 4 to 8 nucleotides. Already told you some are 3 to 4, 4 to 5 to 6, 4 to 8. So that is depend on the species, right? So these recognition site mostly palindromic, which correspond to the nitrogen based sequence that read the same backward and forward. Already told you.
Yes, sir.
Right? This is the mirror-like palindrome. Mirror-like palindrome. Already given example also GTATAC and same direction GTAT in your book page number 106 clearly given. Means TATAC same direction GTATAC. Can you read this direction and this direction? Same sequence. It's a palindromic sequence, right? And another important one is here. The restriction enzyme recognition site. If you cut the enzyme sequence, two type of cuts are there. One is sticky ends, another one is blunt ends. Sticky ends means, for example, this is a G. Let me write clear. Whatever it may be, in what according to your textbook, taken from your textbook sequence, but you can write any sequence. GAATTC. Same direction GAATTC, right? The cut the direction to right. Now the two ends separated. One is G C T T A. Right? Is it clear? Hello?
Yes, sir.
Right. So now these are the sticky ends. This red color TTA sticky end. These sticky ends with help of ligase enzyme attached with the complementary another nucleotides such as, can you give? Hello?
Yes, sir.
What are the sticky ends here? TTA sticky ends.
Didn't get it.
Same A T. This is attached to this sticky ends, this empty place. For example, first of all, there is no AATT. Only this is completely separated, G separated, and this direction also separated. Now this is ready to stick. Stick means attachment. Now this region, the leave for attachment another nucleotides. There is some space to attachment, isn't it? Hello? Hello?
Sir.
After sticky ends cut, is there some space to attachment other nucleotide? Okay.
Yes.
So these nucleotides attached with same complementary AATT. Now the bond formation and like a recombinant. Now it is called as recombinant. That means another, for example, this is the DNA, red color DNA. The DNA segments cut with help of restriction endonuclease, and this cutting segment inserted with another foreign DNA with same sequence. Another foreign DNA with same sequence, right?
Yes.
Now the both DNA are combined. That is called recombination. Both are combination. So that's why it is called recombinations DNA. Another one, blunt end formation. Blunt end formation means same. Blunt end formation. For example, C, 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 G, 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 C, C. Cut this direction. Is there any sticky ends? Hello? This is C, C, G, G, G. Here G, G, C, C, C. Is there any sticky ends?
No,
Hello?
sir.
No sticky end, no place to attachment, another DNA, another DNA nucleotides. Isn't
Yes,
it?
sir.
So this is called blunt ends. This is called blunt ends, right? Different restriction enzymes that recognize the same sequence are known as neocytomers. Just wait. Neocytomers means different restriction enzymes. The one enzyme, the two enzyme, three enzyme, different type of restriction enzyme recognize the same sequence. One recognize the same sequence. That means GATTC, for example. Two also recognize the same sequence. Here three also recognize the same sequence. This type of enzymes known as neocytomers. These are called neocytomers. These often cleave the different locals, the different sequence. However, different enzymes that recognize and cleave in the same location is known as isocytomer. Isocytomer means here also same different enzyme recognize and cleave in the same location. The first one only neocytomer, different restriction and then recognize the same sequence but not cleave. But isocytomers means recognize same sequence and the cleavage also same. Cleavage also same means cleavage maybe sticky ends or maybe blunt end. Same it is sticky ends. For example, GAATC cut the same between the guanine adenosine, and 2 also cut the same between the guanine adenosine, and the 3 also cut the same sequence. Here means here the recognize the same sequence and cleavage the same sequence. That's it. That is isocytomer. Neocytomer only recognition same sequence. Isocytomer recognize and cleave the same sequence. That is the different. Neocytomer and isocytomer. Is it clear? Recognition side, hello?
Yes, sir.
Right. Now, next one, type 2 restriction endonuclease. Of course, it is already completed in 1 and 2 and 3 form, but especially this type 2, some more important. Type 2 restriction endonuclease. This type 2 restriction endonucleases mean recognize, modify, and cleave. Cleavage. And already told you they required only magnesium ion as a cofactor. And this is already told you, this is the key involved in RDNA technology, and type 2 restriction endonuclease generally used as a key reagent for variety of application in molecular biology and molecular biology also. Molecular biology and DNA techniques, including GNA mapping, restriction fragment length polymorphism, means RFLP. I will explain later this about RFLP, and DNA sequencing and cloning process. In this respect, they can be considered to the workhorses. Because why we called as a workhorses of molecular biology? Which enzyme called as workhorses of molecular biology? Workhorse. It look like horse. What is the function of horse? Multiple functions. Like this is also involved in DNA technique. Already told you recombinant DNA technique and genome mapping, RFLP technique and DNA sequencing and cloning. This is involved in several aspects. That's why it's called as workhorses of molecular biology. Workhorses is very, very important. Workhorses of molecular biology. Type 2 restriction endonuclease recognize the short nucleotide sequence, very short nucleotide sequence, and cleave double-stranded DNA at specific site within or adjacent to those sequence. Order told you, and when compared with 1 and 3, 1 and 3 recognitions are different and cleavage are different. But type 2, where the recognition site recognized by RNA polymerase 2, there only cleavage also takes place, exactly place and just adjust, exactly place or just adjacent to the recognition. The length of this recognized sequence varies considerably. For example, first enzyme 1, NOT 1 recognition 8 base pair sequence, and SCIU 3A1 recognize only 4 base pair sequence. For example, first of all, NOT 1, this is the DNA strand, AGCT something TCGA. Only recognize these 8 nitrogen bases. Some are 3DGC2 AACT 8 sequence. So this is a recognition site. The length of the recognition sequence indicate how frequently the enzyme will cut. An average random sequence DNA enzyme with an 8 base pairs recognition site will cut every 48 or 65,536 base pair. Means actually this is the recognition site. From the recognition site, it may be cut the number of nucleotides. Only recognition site nucleotide number may be 4 or 8, but cutting region more than that. More than that means this is only 4. But after recognition, the start from here and ended somewhere after 48 nucleotides. Minimum, maximum 65,000 base pairs. Such a long DNA nucleotides would be cut by restriction 2. For example, if you take 4 base pair recognized site, will cut every 256 base pair. Every 250. For this is entire DNA strand. 1, 2, 3, 4. This is the 4 nucleotides. And after recognition, somewhere here 256 base pairs. 256 base pair. This region to 256, this region totally cut. And again another recognition site, and again 250 nitrogen bases cut. Cut may be sticky ends or blunted ends. Is it clear? Hello?
Yes, sir.
Right. The 4 base pair recognition site will occur every 256 base pair. The enzymes with long recognition sequence they're also called rare cutters. Long recognition site. Long recognition site. Enzymes with long recognition site is called as a rare cutters. So for example, I already told you only 4 or 8 recognition nucleotides. If it is behind that more than 8, this is called as a rare cutters. This is called as a rare cutters. That means if exceeded 8 base pairs, that is called rare cutters. And cleavage site can be, I already told you about the palindromic sequence, but no need to repeat again. The cleavage sites can be unambiguous and ambiguous. What is unambiguous and what is ambiguous? Unambiguous means it should be in palindromic sequence. In palindromic sequence, they clearly appear the direction forward and backward. We can clearly identify the nucleotide sequence also, that is unambiguous. Right? What is ambiguous? Ambiguous, not sure. For example, unambiguous means same palindromic sequence. For example, let me clear right. Are you able to see? Hello?
Yes, sir.
First, before that, you know already sequence that is, for example, A, T, T, A, T, for example. The same way A, T, T, and A, T. The same direction. It should be unambiguous, clear. This direction and this direction clear, right? Is it okay?
Yes, sir.
Unambiguous sequence, okay, right? Now, when we see the ambiguous sequence, here the some sequences look like G, A, N, G, A, N, T, C. Can you write complementary sequence to this? Hello? Can you write complementary? Now tell me the complementary sequence for this. G for G, complementary sequence. Hello?
C.
C, T, N, S, G.
Then what about N here?
N.
Here
don't
T,
know.
adenine, and C, guanine.
Yes.
This is not clear. N,
Yes.
it is N sequence not clear. The N representing any nucleotide. And given some examples, if in sequence M is present, M may be adenine or cytosine, but not sure. Anyone among this, between the A and C, maybe A or maybe C, or for maybe A or G, W A R T, S C R G, Y C R T, K G R T, V C G, H C T. Like this, if we take the N here, N refers all nucleotide guanine, adenine, thymine, cytosine. Is this sequence ambiguity or unambiguity?
Ambiguity.
Right, this is the ambiguity sequence. That means the restriction endonuclease to the cut where the ambiguity sequence there also it's cut into two fragments. Right. Next one, a cohesive ends and blunted ends. Already told you. Cohesive ends, blunted end. Cohesive end means sticky ends, nothing but sticky ends. Like this. This sticky ends. If they cut the same like this is blunted end. Is it clear? Blunted and sticky ends. Should I explain?
Yes, sir.
Hello?
Please.
Right. Right. And here the last one, applications. Applications just no need this much of applications. Of course. The main application of the restriction endonuclease recognition and cleavage and modify the DNA. That is the main sequence. And also this is involved in rDNA technology and RFLP. And this terminology I will explain later. Okay. And this application also will be covered later.
Okay,
Right? Is it okay?
yes, sir.
Okay, sir.
Right. Now clear our restriction endonuclease, anti-restriction endonuclease.
Yes, sir.
Now 8th unit modifying enzyme, I will tell you later about the ligases in the rDNA technology and recap protein also no need. Cloning vectors also I will explain later. Genomic cDNA library also. Okay. Now the polymerase chain reaction. 11th unit. 151. 11th unit. 151. Page number, turn on page number 151. Turn on page number 151. Right? PCR, polymerase chain reaction. The abbreviated form PCR, polymerase chain reaction, is a modern technique. You must have heard about this during the corona time. PCR
Yes, sir.
test, RT-PCR test. So this is the modern technique in molecular biology that is used to amplify a DNA segment. That means separate a DNA, and this DNA makes a number thousands of copies. That is the main the properties of this PCR. The first, it is developed in 1983, Carl B. Mullis. He awarded Nobel Prize also in 1993. So remember the scientist name, Carl B. Mullis. Carl B. Mullis. Right? Let's see the procedure steps of Polymerase chain reaction. Polymerase chain reaction. The first of all, what are the requirement for the PCR procedure steps? Mainly, in orderly, first one, DNA template required. DNA template required, and primers also required, and DNA polymerase also required. DNA polymerase. Mainly these three very important. Template, primers, DNA polymerase. Let's see the procedure steps. The first one, there are three major steps in PCR which are repeated for 30 to 40 cycles. Repeated in 30 to 40 cycle in thermocycler machine. Already given in our textbook 153 page number. The diagram 11 PCR machine which is called thermocycler machine. Now let's see the three main steps. First one, denaturation. First one, denaturation. Denaturation means at the 94 degrees centigrade, double stranded DNA melts open to single stranded DNA. The melting point not exactly 94 degrees centigrade, that is the depend on the species. Remember it. First of all, this is, for example, this is double stranded DNA. When we heat it 94 degrees centigrade or almost 90 to 100 degrees centigrade, but mostly 94 degrees centigrade. At 94 degrees centigrade, this double stranded DNA becomes single stranded, right? Okay. Is it clear?
Yes, sir.
Right. For example, this is the 5.2, 3'. This is 3.2, already anti-parallel direction. Do you know the DNA structure? Parallel, anti-parallel direction. One is 5 and 2, 3 and direction. Another one also 5 and 2, 3 and direction, but backward, back reaction. One is forward direction 5 and 3 and another one is 5 and 3 and backward direction, reverse direction. Right? So the first step completed. Now second step, annealing. Denature completed. Now second step, where I write? Annealing. 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 Second step, annealing. After separation of DNA, the DNA should be attached with the complementary primers. Complementary primers at the 3-hydroxy end. 3-hydroxy group, 3 end at the site of hydroxy OH group. This is 3 end, right? This is also 3 end, right? Okay. Hello.
Yes, sir.
Only the blue colors, for suppose this is also primer and this is also primer. These complementary primers attached to at the 3 direction and at the this both 3 direction. And this is carried out at the temperature 54 degrees centigrade. At the 54 degrees centigrade. Only 54 degrees. This is very important for long question. Please careful. right? Right. So now, what is the function of primers? The primer just identify the complementary sequence, and next it is assisted to the attachment of DNA polymerase. Attachment to DNA polymerase through. So this is only the annealing process. Annealing process only identify the sequence, complementary sequence, and attach it with primers at the site of 3-hydroxy end. Where the 3-hydroxy end is first strand, this is the 3-hydroxy, and second strand, this is the 3-hydroxy. Now which direction the that means the next step, third one, extension, right? Extension. Extension, right? First denaturation, separated, annealing, attached with primer, next extension. The extension started after adding with DNA polymerase. After adding with DNA polymerase, right? Let me write here DNA polymerase. DNA poly, here also DNA poly. Now which direction? In which direction this DNA extended? Remember always one thing, all is newly synthesized DNA always 5 into 3 end. Remember this one. Always 5 into 3 end. Right? Now tell me the first stand from right to left. That means the first stand from back direction or forward direction.
Forward direction.
Forward direction, sir. First one.
First one forward direction.
Yes, sir.
Here primer is present, okay? And DNA polymerase attached here. How can you move forward? Okay, right. Forward means, for example, can I rough?
Yes, sir.
Five and 3 end. Antiparallel direction. Another one also 5 and 3 end. Actually, these two directions. Already told you newly synthesized DNA. Newly synthesized DNA 5 and 2, 3 end direction, right? Now, for example, this is the template 5 and 2, 3 end template. Right? Now tell me on the 5 and 2, 3 end template DNA stand, the direction of extension. Backward or forward?
Forward, sir.
Right, as you said, forward. So this is the 5 end. The 5 end opposite should be 5 end opposite. What is the anti-parallel side? 5 end opposite.
3 end.
3 end. As you said, this direction 3 end to 5 end, right? This is the forward direction. I'm writing and the giving the line for forward direction, right?
Yes, sir.
Okay. Is this forward or backward?
Forward, sir.
Right. As you said, the forward direction 3 end to 5 end, right?
Yes, sir.
3 end to 5 end. What I'm said just earlier, the newly synthesized direction should be
5 to 3 end.
5 to 3 end. But is this 5 to 3 end?
No, sir. No.
Now tell me the direction backward
It
or
is a backward direction.
forward.
3 is to 5.
Right. Backward direction, right? 3 is to 5. Already the 3 end present on the 3 end. The primer attached here. Primer attached here. Sorry. Primer attached here. Polymerase also attached here. Now it is started 5 end. 5 end to backward direction. It is 3 end. Right? So now synthesize 5 end to 3 end, backward direction. What about the second stage? What about this stand? This the primer attached. DNA polymerase also attached here. Now synthesization direction, forward or backward?
Forward
It is
direction.
3 end. Right, it should be forward because the opposite 3 end, opposite 5 end. Now it's move
Yes.
forward. Right?
Yes, sir.
Is it clear?
Yes, sir.
Now tell me extension direction. Let me clear. Just wait. Already two separated stand. For example, this is 5 end to 3 end. Opposite direction 3 end to 5 end. Here primers. Here also primer attached. Now tell me this direction. DNA polymer attached here, direction backward and forward. The first line.
First line backward direction.
Right, backward like this.
Yes, sir.
Like this. Right?
Yes, sir.
And finally forms the another DNA. And same here. This region here, starting here.
Direction.
This forward direction like this. Finally forms the DNA strand. Now fully matured DNA, two double-stranded DNA form. You take one DNA, it becomes two for one cycle. The same cycle repeated in thermocycler mission. Now, how many DNA strands are formed? Second
Two.
cycle. Total number. Actually, first cycle, for example, this is the first DNA. From the first DNA, for 1 and 2 DNA strands are formed, right? Hello?
Yes, sir.
Is it clear?
Okay, sir. Okay, sir.
Don't get confused. I am starting with one single stranded DNA.
Okay, sir.
Sorry. One double stranded DNA. One DNA strand like this. One DNA strand. After completion of denaturation, annealing, extension, it becomes 2 DNA. This is one DNA and this is second DNA, right? Now this both enter into the same process. Then how many DNA strands are produced?
In second cycle.
In second cycle, how many?
This 2 becomes 4.
Right, 4. Then 4 becomes
8.
8. So the simple direction 2n. N for cycle number. N for cycle number. Okay. For example, the DNA after completion of 16 cycle, how many DNA strands are produced? 2 into 16. Like this 2 into 2 into 2 into 2 into 16 times. Then what? That is the answer. Okay.
Yes,
Right? Is it clear?
yes, sir.
Yes, sir.
Any doubt in this?
No, sir.
If you understand about this procedure step, you can understand next RT-PCR, reverse real time and reverse transcription PCR. You can understanding this both. That's why I should clear in this. The simple 3 steps again. First one, denaturation means suppression of DNA at 94 degrees centigrade. Second, annealing, 54 degrees centigrade. Annealing means simple attachment of primers. And third one, extension. The extension occurs after attachment of DNA polymerase enzyme. The DNA polymerase enzyme, one move backward, another forward. After that, 2 DNA strands are formed for one cycle. For each and every cycle, the DNA number would be multiplied. Right?
Yes, sir.
Okay.
Okay,
Right.
sir.
So this is PCR steps. This is very, very important in theory exams as well, practical also. Right. Was it clear?
Yes, sir.
Any doubt in this?
No, sir.
Right. Next, again, join in plant molecular biology second session. Just wait. Right. Meantime, just see this. See this. Polymerase chain reaction step procedure. First one, this is the DNA step. This is DNA double stranded, right? Step 1, denaturation. At 94 degrees centigrade, this double stranded helical unwind like this. This is 5N to 3N and this is 3N to 5N, right? Annealing, second step, annealing 54 degrees centigrade. At 54 degrees centigrade, the primer attached here, 5N to 3N direction here, this is the primer, and this is also 5N to 3N direction, this is direct. One is reverse, one is forward primer, right? Extension after polymerase enzyme, DNA polymerase, it another name TAC polymerase enzyme. The blue color is TAC polymerase enzyme. One is backward direction, another one is forward direction. The extension carried out at the 72 degrees centigrade. Is it clear? Note down this diagram.
Okay, sir.
Okay.
Okay, sir.
Please join again in 3 o'clock. The next session, last the course 8.